Well, it's a real pleasure. Thank you very much for having me. I'm excited about presenting this topic of what lies ahead in terms of digital twins for precision medicine. I do have a talk on YouTube that you can see, and in a funny way, I'm multiplying. So one of the students in our lab did this, but I go over some history of digital phantoms and the future of digital phantoms, which is going to be digital twins, which are going to be much more realistic. So you can take a look at that, but I will convey some of the messages there and here as well. So we've had different generations of phantoms or anthropomorphic digital models, and they've been improving in terms of realism, but nowhere close to where we want to be, and that's the point I would like to make. So the first generation, essentially, you see geometric shapes. And again, we've been moving from simple to complex and to more heterogeneous and more realistic structures. And you had the models where you would have segmentations of different organs, but they were very voxelized. And finally, you move towards more, using nerves or different techniques in graphics where you would be fitting continuous surfaces to the structures. So they would be really multi-scale, and you could define them at different resolutions. And I think what we're moving towards now is making these phantoms personalized. And so therefore, in a sense, they're going to be digital twins, which means phantoms that are personalized to individual patients, not just a generic population of patients, but you and you and you, each one of you will have your own digital twin kind of idea. So again, in the past, these phantoms have been used for different purposes than what we want to use them now. And they've been used for example, simulating virtual scanners, and to, for example, for optimizing designs of future scanners, or for looking at different imaging protocols to see which protocol is a better protocol. So you could have a virtual scanner, a virtual patient, and even a virtual observer to narrow down the list of protocols you want to use, and then finally you present a few of those protocols to clinicians or radiologists, and they're going to pick one of them. Kind of idea. So this idea of virtual clinical trials has been mentioned, right? So these three things where you have a virtual population, virtual scanner and acquisition, a virtual interpretation to narrow things down significantly. And here's a example of a population by my friend and colleague, Dr. Paul Seegers. Both of us used to be at Johns Hopkins University, and he's at Duke right now. So you could have a population of patients and try your methods and techniques on them. And again, you could be looking at different reconstruction methods, different algorithms, and pick the best ones, for example. You could also do things like retrospective dosimetry, where you would have a series of, imagine you're doing a CT scan on this series of population of patients, virtual patients, and you could analyze, how much dose is everybody getting. But you could also do it prospectively, meaning when a patient shows up to the clinic, you would find that scanner, that phantom digital anthropomorphic representation that best matches them in terms of, for example, geometry, shape, just size in general, or whatever criteria that you have for matching. And since you've done extensive calculations, for example, to do dose for different protocols on the digital version of that patient, then you could then anticipate and predict how much dose they will be getting, and therefore pick very proactively which protocol you're going to use. So this is as close as we've gotten so far as digital twins in that sense, where we have an atlas of patients, and you would probably be matching the patient with one of those individuals. But this is, in a sense, but again, here are other examples of matching that can be done and profiling. But in a sense, this is easy relative to what we want to do, because we're talking about here just looking at the anatomy of patients, and these models have nothing to do with the physiology of the patient, their biology, and the details of, for example, let's say cancer. So we need a lot more things to happen. For example, with the existing phantoms, we cannot predict if you do chemotherapy on the patient, how much is going to go where, and how it's going to treat the patient, or things like targeted therapies. I'll talk about radiopharmaceutical therapy, where you inject a radioactive material into the patient, it goes to different parts. So can we use these phantoms to predict how much therapy will be happening to the different targets in advance, so that we could optimize the therapies? So can we do virtual therapies? That's the idea, virtual therapies, to optimize what we're going to do for this patient and then actually use that on that patient. So the concept of digital twin, therefore, is going to be at the center of personalized and therapies. So what is a digital twin anyways? It's a numeric representation of a patient, and it could span even one's entire life. It can be updated with real-time data, for example, cumulative radiation dose that they're getting, reflecting history and current conditions. So digital twin also can be aging and can be changing with time for two reasons. One, you're getting more information about the patient, and two, the patient is actually evolving. And so this can be used for virtual therapies and simulations to aid physicians in complex treatment planning scenarios. And again, by complex, we're not just talking about, we're talking about a collection of things, not just external beam radiation therapy, which in a sense you could argue, is being done well right now, because you have a nice model of the patient, CTCM or whatever it is that you're using. But again, we're trying to do this for complex therapies like chemotherapy, radiopharmaceutical therapy, where you're injecting, or drugs that are being injected into the patient, what's going to happen to the patient. So here's a nice article in Nature Medicine that you may want to check out. Digital twins for predictive oncology will be a paradigm shift for precision cancer care. There's movement in this space, and the concept of virtual physiological, blueprints for patients and twins for patients. So some papers are starting to refer to this. So here's just one example. This is for immune digital twins, but the concepts are general. So this is a workshop on multiscale and multimodal imaging. So you're going to need multiscale data at the body, organ, tissue, cellular, molecular levels. You're going to integrate these, and these could be captured by multimodality imaging and also non-imaging information. For example, these will be integrated and to develop in silico simulations on which you can do virtual clinical trials, and therefore personalize what you will be doing, especially on the therapy side, therapy optimization. But it will be also used to diagnose better and to predict better. Here's another example. For example, we've got imaging happening. What I'm going to keep emphasizing is that an image is not really operable. You may say, well, of course it is. I can flip an image and I can do stuff on an image. No, but I mean it in a very clinical sense. When you have an image, there's not much we can do to it compared to a digital twin. In a digital twin, you could do virtual treatments. You cannot virtually treat an image because it's an image. You don't have relationships happening. But if you have a digital twin, I could say, oh, if I inject this much at this location, how much is going to go where? You cannot do that to an image. So you're going to have some images. So images are starting points, not endpoints. And then you could do, for example, mechanism-based modeling, where you're looking at things like tumor growth or tumor transport phenomena, where, if a drug is injected, what are the processes by which it could, for example, diffuse through extravasation, enter tissue and diffuse, etc. Microenvironments, therapeutic response, etc. You could be using ordinary differential equations to link the various organs together. You could be using partial differential equations to model both change as a function of time and space, for example, diffusion, as I mentioned. So using these, then you could have models. Again, these are not just images. You could have models that sort of, those models understand and derive and extract, and using inverse methods, extract things of importance for the patient, and like it could be the density, permeability, put a holistic picture of the patient together, and now you could do experiments on this and predict, tumor response to treatments. So just to show you another example where you could be doing pre-therapy imaging, maybe use that on its own, but also you could do some initial treatment and do some monitoring imaging, take all this information to build a patient-specific digital twin. And based on this, you could say, okay, I can think of maybe 3 different plans, A, B, C. Which one's going to do? Let's run these. Let's do a virtual clinical trial, this virtual therapy. And you're going to be like, oh, plan B is better. So let's now do plan B instead of plan A or C. And this is constantly updating itself with new information that you're getting, and also as the patient is evolving. So this is a workshop, again, multimodality, imaging, and multiscale. So there's going to be a multiscale source of things, multimodality. You could be, to build digital twins, you could have experiments done in vitro, you could have animal models, human data, with different levels of sophistication and scale. And you're going to need multi-fidelity modeling too. And I'll get to this later too. When we do AI, multi-fidelity cost functions for AI are going to be important, where you're not just going to try to fit the data, but you can also enforce the biology you have. So I don't know if you've heard of it, physics-informed neural networks or biologically informed neural networks. I'll get to that in a second, where the neural networks are going to be informed by the biology. So again, you have a new patient with some data. Based on that, based on the imaging or non-imaging data that you have from the patient, you do some kind of an inverse problem to estimate the parameters in the digital blueprint. So I'm going to call it digital blueprint versus digital twin. A blueprint is like a generic model. But once you take the parameters in that blueprint and personalize it for this patient, then that blueprint becomes a twin of that patient. So a blueprint is just a general generic model, whereas a twin is something personalized for you because it's a twin of you. So you take the blueprint that you have and you estimate these parameters in it, and that gives you a digital twin. And now you could feed into that digital twin the treatment plan that you have and different design parameters, and based on that, you could anticipate what will happen to the patient and therefore pick the best therapy before you do a real therapy. So here's just another example, a European consortium, and again, these consortiums are starting to pop up. I think what they really are often are major grant-seeking efforts that have become successful, and now they have pitched these ideas, and now they're moving in these exciting directions. So here's an example in the brain, the neurotwin they call it. You can check this out more here, where you would have a real patient. You could do all kinds of imaging on the brain, for example, fMRI, what have you, PET, EEG, etc. Based on these imaging information, you could build, and also again, non-imaging can be done too, and should not be neglected. Based on that, you build a neurotwin, and on that neurotwin, you could do different things, right? You could ultimately come up with an optimal stimulation protocol for therapy, for example, of Alzheimer's disease. And once you optimize that, you try it on the patient, doing, for example, new imaging, new information, you could go back. So it's a loop. And we're also in our group, we're proposing this thing we're calling Theranostic digital twins. So I'll go over that as another example. I'm giving you a different example. So I'll give you one for immune digital systems, one was for neuro. We had like breast. These are just examples, and I'm going to talk to you about Theranostic digital twins. So this is an excuse to share with you our excitement about the field of Theranostics, if you haven't heard about it. So Theranostics is composed of the two words diagnostics and therapy. And the idea is that if you have the same target that you use for imaging and therapy, it allows new possibilities. For example, imagine a cancer cells, a tumor with cancer cells expressing a certain or creating a certain protein. If you design a molecule that binds to that protein, right, that has been expressed by those cancer cells, well, if you then radio-label that molecule with something like fluorine-18 that emits light, well, you could do PET scans on them, okay? So because it's a molecularly targeted imaging of those cells that are being expressed. It could, for example, prostate cancer. In metastatic prostate cancer, the cells are all over the body, for example, and you could image them all over the body. Now imagine instead of radio labeling with something that emits light and gamma rays, instead you radio label with something like yttrium or lutetium. Instead of emitting light and gamma rays, it emits particles. So then they sit on these cancer cells and they bombard the heck out of them with these particles, breaking, doing DNA breaks, etc. So this is Theranostics because you're imaging what you're about to do, or you're treating what you have seen. So treat what you see and see what you treat. You could first see what you're about to treat. You could treat the exact same thing you just saw because they're the same target, and then you could monitor therapy and see what you just treated. So again, I'm just showing you the exact same thing. You have a molecular target, okay, and let's say a protein that has been expressed in a cancer cell. Then you design a molecule that binds to it, and you radio labeling it with something that can emit light. That gives you imaging, like a PET scan or a SPECT scan, for example. That can happen, a PET scan. And then what you have, you get this kind of imaging. And then if you radio label it with something that emits particles like electrons, beta minus, it could also emit light at the same time, but this is doing the therapy. And if it is emitting light at the same time, that's even better. You could actually see as these tumors are being bombarded, you can actually also get some light out of them. You could actually image them. So there's been miraculous, developments in this field of, we call it radiopharmaceutical therapy. It has different names, radionuclide therapy or radiopharmaceutical therapy. But look at this cancer patient. This is a neuroendocrine tumors. This patient could be hopeless. There's really nothing to do for this. You obviously cannot do external beam. It's all over the place. But you inject these molecules, radio-labeled molecules, they go and the body delivers them wherever those targets are being expressed. And after time, things will improve. Now, this one is an example of a modest improvement, but there are examples where there is a tremendous improvement. There's an example of prostate cancer, metastatic prostate cancer. No therapy is working. The patient is castration resistant, which means hormone therapy, nothing is working. So this patient has no solution. There's no therapy for this patient. But radiopharmaceutical therapy has shown up, and look at what happens to these patients before and after therapy. And again, it's beautiful because the body is delivering these radiopharmaceuticals to these tumors, and basically you're seeing these kind of things. So now, they have really shown very good results, but the problem is that these clinical trials that people have been running have not been personalized. Everybody's getting the exact same dose, and we want to change that. With those non-optimal, therapies, people are getting an extended life of about 5 months, which is very good. I mean, sometimes when you get even 2 months, that's big news. But these are 5 month, or 4 month overall survival improvements, 5 month in progression-free survival. And these are the kind of curves you get. But sometimes, in one of the trials that we see, initially it's good, but then later it comes back. Now, can this be improved? And we're pushing back. We're pushing back against people running clinical trials without optimizing therapies. Everybody's getting the same dose. One size fits all kind of one. We're saying, no, you got to have digital twins of patients and change how much you inject to different patients. So the idea of diagnostic imaging using these theranostic techniques has so far been to select which patients express those cancer cell targets and then do the therapies on them. We're saying, no. This should be done not just to select who's going to get that therapy, but also to optimize how much therapy they get. Okay, so digital twin is can look at different radiopharmaceutical therapy approaches, changing how much we inject into a patient, where we inject into a patient, how many times we inject into the patient, etc. So that's the concept of Theranostic digital twin. In the interest of time, I'm going to move a little bit faster, but ultimately what we're trying to do is we want to optimize towards maximal impact on tumors and minimal toxicity at organs at risk. But to do that, we need to improve biological models. So again, you need multiscale modeling because in time and space, cancer is spread. So again, cancer is insufficient. A scale-specific, a scale-limited model is insufficient to cover cross-scale mechanisms. Again, cancer is a class of diseases that is multiscale and expresses the cells in a multiscale sense. So here's an example of a model, very simple model. It's not that simple, but looks kind of simpler than the typical model. These are physiologically based pharmacokinetic models where different organs through ODEs, ordinary differential equations, are linked to each other. And I guess personalization would mean to find all these rate parameters for individual patients. People have used these models in the past to get population level understanding, but increasingly the idea is to use them for personalized digital twins. So here's a model we've built with about 20 or 19 organs here, and there's a lot of details happening here. There's roughly 100 differential equations happening here, because you got, let's say, 20 organs, but each organ you also have multiple compartments in it: vascular compartment, interstitial volume, when you bind to the tumor, when you get internalized, kidneys is quite complicated, etc. So I won't go through these details, but based on that, we could experiment with the purity of the radiopharmaceutical that we're injecting, how much we're injecting, and predict how much dose is going to be delivered to the tumor. So these kind of models allow you to predict the dose that will be delivered to a patient. You could also model tumor transport phenomena, things like the micro, like things like diffusion, convection, etc., and then look at microvasculature and the numerous problems. So now I want to talk to you a little bit about the AI angle to this. What is the AI angle to digital twins? At the end of the day, what you have is a lot of data, multi-scale, multi-modality data, and you want to build a digital twin. Can AI help us with building digital twins from all this data? So there are some review papers. This is one example where they talk about how machine learning and multi-scale modeling go together. You can sort of look at this diagram. I don't have time to go through it, but there are relationships between multi-scale modeling and machine learning and how they can support each other. But just to give you one example, here's a work on mathematical oncology. So there's different names for this field too, right? Computational oncology, mathematical oncology, and people have come from different angles to this. And in our field, we're trying to do computational nuclear oncology because we're in nuclear medicine, but I'm trying to present in a very general sense. So you could have patient data. You could have a mechanistic model that includes, let's say, diffusion and, for example, binding, etc. Well, AI can help in different senses. First of all, to actually fit the data to the mechanistic model, you could use AI to augment sparse data. That's one angle. Another way, so you can go this way, that way, or that way, this way. Another way would be you could use AI to get an initial condition, and then you solve the mechanistic model. They can sort of help each other out. I'll show you in a second that you could use neural networks for the inverse problem, but they could be biologically informed neural networks. So you could actually write a neural network to solve and personalize a digital twin, but with guidance from the mechanistic model. So you're not just doing pure data fitting, but you're actually getting guidance, and you're having penalty terms that if you're not observing the mechanistic model, you penalize the fitting. So the problem with AI approaches is that they often don't incorporate the underlying physical mechanism, right? They're kind of blind to black box issue. And as a result of that, these things are starting to emerge. Physics-informed neural networks, biologically informed neural networks, systems biology-informed neural networks. These are very, very exciting frontiers, actually. So maybe I'll quickly go over that concept. So, we've heard of the universal approximation theorem, which sort of basically says, if any reasonable function with a sufficient size, depth, or height of a neural network, you could discover it. But the problem is that the universal approximation theorem is all about the approximation error. It says if you have a sufficiently deep, for example, network, you could fit. But it doesn't really talk about trainability and generalizability. It sort of says you can derive it. But if a model is too deep, right, and has too many parameters, trainability is an issue, and generalizability could be an issue. To tackle that, people already have done something. For example, convolutional layers have translational invariance kind of built into them. So we're sort of already saying if a dog is shifted, if a dog is here or here or here, it shouldn't matter, it's a dog. So this spatial invariance is already been built into these things. Why can't we build more invariances into our neural networks? right? Because again, by design, this has been built into it. Well, why can't we build nature's laws into neural networks? Okay, so things like there's been from different angles. When you have lots and lots of data, perhaps you don't need any physics. You just discover. But if you have limited data, you could actually get you could get help from the laws of physics and biology and nature to fit your data, make it more generalizable. Even when you have a lot of data, they can make the models easier to train with less parameters, but especially if you have less data, which is a reality in medical imaging, they can improve generalizability and regularized training. So the main question is how we can incorporate physics and biology into learning, right? So here's an example of physics-informed neural network. One of the key insights we just had the inventor of PINS present to our group, Dr. Raisi, and he said one of the key insights he had when he developed this was that the derivative of a neural network is a neural network. That was the key insight. The derivative of a neural network is a neural network. So you have a bunch of derivatives in, for example, let's say you want to enforce this law. It could be anything. It could be, Schrodinger's equation or in physics, or it could be like some law of diffusion in biology. We tend to have derivatives there, and therefore you have neural networks. You can take the derivative of a neural network. That gives you another neural network, and you could fit these, and you could enforce, or you could enforce these as penalties, so that if you're not observing this law, you're going to penalize the fitting. So again, you might have, and this is another work, beautiful work. You may want to check it out. This is biology informed neural network. We also had John Lagergren present to our group recently, and again, so you have a neural network. You could have certain laws of diffusion, etc., built into it. Take all these derivatives and enforce in some way that biological rule or that system that you're having. Now, how do you actually incorporate these things? Well, again, so far we haven't been, right? We've just been doing some kind of a loss function that just has a fidelity term that just fits the data. But another technique perhaps would be to have a neural network architecture that as a hard constraint enforces the laws of nature. And this appears to be an open problem. How do you enforce a law in a hard constraint in a neural network? That seems to be an open problem right now. And it's a difficult problem. That this has to be observed. But if you enforce it as a soft sort of a penalty, right, as a soft constraint, this is what people have done, right? So you add to your loss function another term that says, hey, you gotta follow this rule, or else I'm gonna penalize you. It's a soft constraint. Okay. So these are just examples of frontier research that is going to be happening. Digital twins, in the sense that I'm talking about, is very new, and it's a frontier research, and I think it's going to define the career of many people. So that's why we're excited about it, and we're constantly talking about it. And I think, again, we're specifically pursuing it from the angle of molecular imaging and therapy and theranostics, but there's many, many different angles that people are looking at it and should be looking at it. Here's another thing. How are we going to store digital twins? Because we think about images, right? And DICOM was invented to deal with images. How are we going to deal with digital twins? How are you going to store digital twins? Because they're not just images, they're relationships, like those ordinary differential equations, all of this stuff. How are we going to share? How are we going to anonymize, right? Because if a patient's going to have his or her own digital twin, well, it needs to be anonymized, right? In the sense that images are anonymized, for example. So there is this format, the Systems Biology Markup Language, SBML, which is based on XML, that can encode these relationships, right? These, for example, the ordinary differential equations. MATLAB has its own thing, and Python various things. You may want to check out this webpage. This webpage has a biomodels webpage has a lot of. It's a venue for people to share their. These are not digital twins. These are really blueprints, like in general models that people may have for whatever system that they're modeling. So it's not quite a digital twin. But it's a way to share models between different people, share blueprints between different people. But once they're personalized and deployed in the clinic, they become digital twins. So in our own group, we have this software, it's called Raven, that we're trying to use to combine AI model management and digital twin capability. The reason we developed Raven is that, us as researchers, we do stuff on the AI frontier, and then the clinicians are here, and there's this big gap between us, this big wall between us, because I can show you what I'm doing, but he or she wants to see what you've done on their own workstation. So this is a technique where you can take stuff from the DICOM query, your AI, for example, segmented tumor, push back so they can look at it on their own clinical workstation, what this tumor delineation that you just did, what this AI-based segmentation that I just did actually looks like in real life practice. So on top of that, we're enabling this to do digital twins. So for example, here, many parameters are required for these digital twins. They can be extracted from images using some kind of an AI. So you would have the DICOM input, maybe some kind of automated segmentation. You could also have different things extracted and then personalize, the inverse problem, and you arrive at a Theranostic digital twin like a PBPK, or we're calling it PBRPK, physiologically based radio pharmacokinetic model. And then we sort of do that. We move in that direction. So lead project here is Ivan Conclusion from our group. You feel free to reach out to him if you have any questions or you're interested in collaborating. So I'm going to wrap up now. So what the future holds, I think, is going to be multimodal, multi-scale imaging combined with other non-imaging data, aided by perhaps AI in terms of doing the inverse problems. And here's a verb we're using. The verb is to digital twin someone. So digital twinning of patients. We're going to see virtual treatments to optimize delivery of healthcare. Digital twins are going to be used for predictive modeling. For example, a model can be personalized based on pre or intra-therapy imaging and constantly updated. As I mentioned, digital twin can age, can change, can be updated, and different treatment strategies can be explored. Corrective strategies on top of the primary, the primary treatment, like adaptive dose planning or adjuvant therapy, systematic strategies, etc., can be explored. I think we're going to see, hopefully, the increasing recognition that images are not operable clinically, but that digital twins are, and the belief that images are not the end, but only the beginning. So thank you very much. So just to, actually, I'll say one more thing. I think the future is really bright if we let it be, if we sort of daydream and envision a future where we're going to be making much more use of images for personalized therapies. And again, thank you very much.
Thank you so much for the great keynotes today. And yeah, I saw people are raising hands.
Yeah, I think that's a great question. I think, it's funny how whenever a new technology shows up, everybody becomes very, including myself, we become very idealistic, whereas the comparison often is doing nothing. So we're saying, let's do something. Let's do some virtual treatments on patients and see what we see. And I see this in many different examples where you come up with a new way to quantify something and everybody says, well, you're not doing it the best way possible. But hello, I mean, nobody's even doing that. So I think we don't need to be overly idealistic about segmentation methods. I mean, of course, this conference, I think they're the fourth of all the efforts around AI-based segmentation. I think this field is moving very fast. And so I think I'm not worried about that, frankly, too much. I think we're moving fast towards automated quantification of images, segmentation of tumors, organs at risk, calculating tumor burden, measuring perhaps certain important features, radiomic or texture features about tumors, etc., feeding those into our models. So I'm not, I think we should be starting to build digital twins now. And I think as we build them, and it's a bit of a chicken and egg too, because once you build a model, you realize, to really estimate this parameter, I need this kind of imaging. So by building digital twins, you're going to actually start asking new questions. It's a paradigm. it's a new paradigm because it's not just about a specific solution. It allows you to ask different questions. For example, in our therapy so far, we've only been focusing on PET imaging. But because of digital twins, we're also starting to think, well, no, I think we need to do CT or MR flow imaging, perfusion imaging, because I really need a way to know what is the perfusion of different tumors. So just because I want to build these digital twins, I'm now starting to ask questions and seek new things that are needed to be measured. Yeah. Thank you. I think there was a question. There was a hand raised at home, but I don't know if I don't see it anymore.
Yeah, any other questions from the audience? Yeah, actually, I have a more general question for yours. That
Sure.
is, which part do you think is the current bottleneck for really making the digital twin for healthcare useful? Because we have already seen that the concept of digital twins has been very widely applied in the industrial management and engineering,
Yeah.
for example, manufacturing. So what do you think is the current key challenge? Is that relying on the data or the model or our understanding of the procedure or something else?
Yeah, that's a great question. You know, the debate that we had at MICCAI on AI, it also revealed some interesting points, right? I mean, there's a resistance in general in healthcare when you try to do things differently. So there's a bit of that. It's like there's so much that can be done, but it takes a cultural shift, and so that's one of the issues. But you know, to your point, you know, in terms of bottleneck, I think one of the big bottlenecks that I see is the fact that these digital twins do have a lot of parameters in them. So just to show you, here's, you know, I mentioned to you, let me show you. Here's a model that we built. You know, you could have something like, you know, 60, 80 differential equations, each with their own rate parameters. So you have a lot of unknowns. And so the question is, all these unknowns, all these rate constants, which one of them do we take from the population? Which one of them do we actually personalize? Because there are just too many things, too many unknowns, if you want to personalize all of them. So I think it starts, this is one of the bottlenecks, is to figure out one of the key bottlenecks is that physicists and biologists, for example, are in totally different worlds, and computer scientists. The point is that, you know, I remember when I was, you know, 20 years ago, when I was doing simple phantom experiments, you know, my wife, who's a biologist, she used to make fun of me. She would be like, "Oh, Arman, I know by tomorrow you will have your simulations ready and you're going to be able to submit an abstract to a conference. But I'm dealing with real life, real nature. I don't know what's going to happen to my mice and stuff." I think in our training, we're often having very simple models, but now that we're faced with reality and we have to build models that are truly replicating what the patient is doing, we have to understand biology a lot, and many of us don't, you know, coming from engineering, physics, you know, computer science. So the multidisciplinary nature of being able to build a digital twin is another thing. So the knowledge gap that we're having that we have to fill in. So there's a lot of learning that I'm doing and people in my lab are doing right now. We're trying to, for example, hire people with systems biology background in our lab, and it's a tough one, right? Because typically, you know, what's a biologist going to do in my lab? That's a mindset. But we want to have those people, right? So bringing people together, making it really, really multidisciplinary is a challenge. And again, imagine, you know, there's 100 parameters here, and I don't want to, you know, spook people, but we do need to understand these more and more and more. And so which one of these can we say reliably we can take from the population? Which one of these do we need to personalize? So we just need more and more knowledge. We just need to be more courageous as a community and go deeper into understanding, you know, the human body and physiology and things like that. So I think those are some of the bottlenecks.
Yes, thank you so much for your explanations, and bringing people together from various backgrounds is exactly what this workshop is aiming at. So if no other questions from the audience, let's thank again for Professor Arman Rahim